Good morning, sir. We are the reporters today. The topic that we discuss today is the aspect of gene therapy. Medical science has detected many human diseases related to defective genes. This type of disease are not curable by traditional methods like taking readily available medicines. Gene therapy is a potential method to either treat or cure genetic related human illness. Yung um, medical science ay meron daw po silang meron daw po silang nakitang sakit na sa ating mga genes na hindi nagagamot ng medicines. Nang nakakagamot ng ay ang gene therapy. In 2015, a team of researchers at the Harvard Medical School and the Boston Children's Hospital stated that they were able to restore basic hearing in genetically deaf mice using gene therapy. Noong 2015 daw po ay merong mga researchers galing sa Harvard Medical School and Boston Children's Hospital na naibalik nila um, yung hearing, yung pandinig ng isang mice gamit ang gene therapy. Human gene therapy was actually first realized in 1971 when the first recombinant DNA experiments were planned. Na-realize daw po yung human gene therapy noong 1971. The basic process. These are process approaches to gene therapy. These are the following. Replacement of mutated gene that causes disease with a healthy copy of the gene. Inactivation of mutated gene that is functioning improperly. Introducing a new gene into the body to help fight disease. Ito daw po yung mga process na ginagawa sa gene therapy kapag isasagawa ang gene therapy. In general, a gene cannot be directly inserted into the human gene or cell. A gene is inserted into another gene using a carrier or a vector. Sa pangkalahatan daw po yung gene ay yung gene na i-insert daw po ay hindi daw po ini-insert sa katawan natin. Ini-insert daw po ito sa isang carrier or vector bago i-insert sa ating katawan. At present, the most common type of vectors are viruses that have been genetically changed to carry normal human DNA. Viruses have evolved a way of encapsulating and transporting genes to human cells in pathogenic matter. Two types of gene therapy. Gene therapy involves the manipulation of genes in cells that will be helpful to the patient but inherited to the next generation. Ang somatic gene therapy daw po ay nakakatulong sa isang patient upang mapagaling ang kanyang sakit, but hindi po siya namamana sa susunod na generation. Yung somatic gene therapy is ini-implant lang siya sa isang cell or kung saan dun yung daan upang gumaling ka sa sakit mo. Next is germline gene therapy, which involves the genetic modification of germ cells or the origin cells that will pass the change on the next generation. Yung germline gene therapy ay kung saan kapag in-inject ka ay sa buong katawan mo, lahat ng germline ay kasali. Pwede siyang mamana sa next generation. Ang next po ay ang magbabasa ay si Rex Rohel.
Stem cell gene therapy. Stem cells are mother's essential from any types of cell in the body. One of the characteristics of stem cells is their ability to self-renew or multiply while maintaining the potential to develop into other types of cells. Stem cells can become cells of blood, heart, bone, skin, muscle, and among. They are different stem cells, but all types of stem cells have the same capacity to develop into multiple types of cells. Stem, stem cell gene therapy. Embryonic stem cells are derived from a 4 or 5 day old human embryo that is in the latest phase of development. The embryos are usually extras that have been treated into clinics where several eggs are fertilized in a test tube, then implanted into a woman. Somatic stem cells are the that exist throughout the body after embryonic development are found inside of different types of tissue. These stem cells have been found in tissue such as brain, bone marrow, vessels, skeletal muscle, skin, and the liver. They remain in a non-dividing state for years affected by disease or tissue injury. These stem cells can divide or self-renew independently, enabling them to generate a range of cells types from a finite organ of a dead. Regenerate the entire original organ. It is generally true that adult somatic stem cells are limited in their ability to differentiate based on their tissue, but there is some evidence to suggest that they can different to become other cell types.
The bioethics of gene therapy. There are ethical issues involved in gene therapy. Some of the increases cited are: 1. How can good and bad uses of gene therapy be distinguished? 2. Who decides which traits are normal and which constitute a disability or disorder? 3. Will the high cost of gene therapy make it available only to the wealthy? Number 4. Could the widespread use of gene therapy to make society less accepting of people who are different? Number 5. Should people be allowed to use gene therapy to enhance basic human traits such as height, intelligence, or athletic ability? Ito daw po yung mga ethical issues na involved sa gene therapy kapag magsasagawa ng gene therapy. Ito daw po yung mga issues na involved. Yun lamang po ang aming topic tungkol sa gene therapy.